FADH2. So these guys, this is the reduced state. This is the oxidized state. So oxidized is without the electrons. Reduced is with the electrons. So glucose and fatty acids are oxidized. Their electrons are moved to NAD plus and FAD to make these guys the electron carriers. And those electron carriers carry those electrons where? To what transport system? The electron transport chain. We're just moving electrons. I know we make it really complicated, but it's all just moving electrons. I wish we could make it simpler.
Dr. Farley, can you repeat that top little portion real quick?
Yes.
NAD and FAD is
So,
which part?
So NAD plus and FAD are the oxidized forms of NADH and FADH2. So these guys are the oxidized forms.
Gotcha.
Because they don't have the electrons. And the NADH and FADH2 are the reduced forms.
Okay.
They're the ones that are carrying now the electrons.
Thank you.
No problem. And so, again, we will revisit, or we won't revisit, but there's still the TCA and the Krebs cycle. So in this case, you're oxidizing pyruvate, which is just the product of glucose, and making acetyl-CoA. And then you're also making NADH and FADH2 during the Krebs cycle. NADH and FADH2. And I have another pen coming in the mail. It'll be here soon. And then you should also remember glycogenolysis and gluconeogenesis and what they are. Breakdown of glycogen, the synthesis of new glucose from non-carb sources is gluconeogenesis. But I promise you, I will not ask you any questions about these pathways on the exam. I am just talking about them because they're leading us into remembering these pathways will help you understand the lipid metabolism pathways. So we're not going to talk about these in detail. But I am going to talk about fed versus fasting state because just like with glucose, they are what's going to determine which pathways are active. So when you're in the fed state, that's going to activate certain pathways like lipogenesis. You're going to create lipids. If you're in the fasted state, that's going to turn on lypolysis, which is going to break down lipids. So again, when you're thinking about how these are regulated, you really need to understand whether the person is in the fed state or the fasted state, and that will help figure out which one's going to be activated. So the same thing, just like with blood glucose, in the fasted state, you're starting to lose glucose. If you didn't eat breakfast this morning or lunch, you are very fasted right now. You're very much in the fasted state. Maybe. Depends on how much glucose it has. But technically, anytime you eat, you're no longer fasted. So technically, if you did drink a protein shake, you're in the fed state. So anytime you consume food, you will be in the fed state. But if you haven't consumed food in the last 10 or 15 hours, you are out of glucose and you are out of glycogen. So you're doing a lot of gluconeogenesis just to keep glucose up in your blood. So your body's probably breaking down various components in order to make new glucose. Yeah?
You're in a fasted state and you're going to exercise, your body will use fat as energy.
Yeah, there is a way when you're working with people with losing weight or whether it's athletes, fed versus fasted states, having a person exercise when they're fasted will help upregulate enzymes that make you a better, quote, fat burner. So yeah, there are ways to, like exercising with it, that it will help increase stimulate certain enzymes that make you a better fat burner. But I would say, generally speaking, unless you're an athlete, I personally wouldn't worry about that, even if I was trying to lose weight. But yeah, there are ways where you can upregulate certain enzymes by exercising in a fasted state. Yeah. So remember, glycolysis frees energy, it's catabolic. Gluconeogenesis consumes energy, so it's anabolic. So the fasting state will increase lipolysis. So if you are fasted, you are going to be doing a lot more lipid breakdown. So lipolysis versus beta oxidation. I'm just going to write it here. So lipolysis is the breakdown of triglycerides. Lipolysis is the breakdown of triglycerides. And what you're doing is you're actually pulling off fatty acids. From the glycerol. Remember, the glycerol is just a 3-carbon molecule. And it's portable. So remember, here's your triglyceride. You got 3 carbons here, and there's 3 fatty acids attached to this glycerol backbone. So with lipolysis, you're just taking off fatty acids from this glycerol group. So you're just going to remove these fatty acids, and then what are you going to do with these fatty acids? You're going to do beta oxidation. So lipolysis and beta oxidation are 2 different things. Beta oxidation, this is something students didn't understand last term, which is why I'm making sure I pointed out, because I could tell on the exam they didn't quite understand the difference between lipolysis and beta oxidation. So lipolysis, you're just breaking down triglycerides, you're pulling off fatty acids. Beta oxidation is when you're taking those fatty acids and you're going to oxidize them. So you're done with the triglycerides by the time you get to beta oxidation. You're just dealing with the fatty acids. And you're oxidizing them, you're taking off taking their electrons, and you're adding those electrons, and you're making FADH2 and NADH. So these guys, you're making these guys. And you're going to make acetyl-CoA. So I'm just giving you a heads up. You didn't even have to write this down right now, but you'll see beta oxidation a little bit here. But beta oxidation is where you're actually breaking down fatty acids to make FADH2 and NADH and acetyl-CoA. Whereas lipolysis is something different. You're getting ready for beta oxidation by breaking down those triglycerides, taking off the fatty acids, and then the fatty acids will be oxidized in beta oxidation. So that's what we're going to talk about in this lecture. So that's just kind of a heads up. And we're also going to talk about ketogenesis in this lecture. That's also something that's going to happen when you're in the fasted state. You're going to do a lot of ketogenesis. Even though ketogenesis is anabolic, you have to build the ketones. It doesn't take a lot of energy, and so it is going to happen during the fasted state because those ketones are going to replace glucose for a lot of your tissues. So you make ketones in order to conserve glucose. And we'll talk about ketogenesis more when we get down to it. Yeah.
There was a quiz question last week about reducing equivalent to fatty acid synthesis. You said that it was NADPH.
Yes. So NADPH is going to be a molecule that's going to be reduced to make fatty acids. So right now we've been talking about fasted state and breaking down fatty acids. But when we build fatty acids, so when we break down fatty acids, we still have NADH and FADH2. But when we build fatty acids, they're going to be the reducing molecule that's going to, NADPH is going to be the reducing molecule that's going to allow us to build fatty acids. So that is true. That is a correct question. It's just, and we will talk about that in this lecture as well. All right, so insulin and glucagon, just like they control glucose, they also control these pathways for lipids. So it's not just for carbohydrates. Insulin and glucagon will stimulate or inhibit lipid pathways as well. And as we know, insulin predominates, same word, in the fed state. Glucagon is the one that's going to dominate in the fasted state. Which means here is insulin over on this side. So you're going to build stuff with insulin. You're going to take up glucose into muscle to make glycogen. You're going to be doing lipogenesis, which is the production of lipids. You're going to make glycogen. You're going to make proteins in muscles. You're also going to be doing glycolysis. In the fasted, and insulin as well is going to stop these guys. So these are going to be what's going to dominate in the fasted state. So in the fasted state, it's going to be lipolysis. So insulin is going to inhibit the pathways that dominate in the fasted state. So it stimulates the pathways that are in the fed state. So these guys are the fed state. And it's going to inhibit the pathways that dominate during the fasted state. And it's the same thing with glucagon. And keep in mind, insulin and glucagon exist as a ratio. It's not one or the other. It's just one will always be dominating depending on how fed or fasted you are. And that's why I have this slide here. Glucagon in the fasted state is going to dominate. It's a bigger term here, whereas insulin in the fasted state, it still exists, but it's just not going to be a lot. So glucagon is going to be the main driver in the fasted state, and this is what is going to turn on as well as lipolysis. So you're going to break down glycogen, you're going to make new glucose, you're going to make new ketones, and you're going to break down lipids. And I could also add here beta oxidation. Because technically it is not like lipolysis and beta oxidation are technically two different things. But all of this is going to dominate in the fasted state. Oh, yes. So proteins can be broken down too in the fasted state. So, and you're going to be making ketones in the fasted state. So, yeah, you could add that as well to this. Oh, I already have ketogenesis. But you're also going to do, you're probably going to be doing some proteolysis as well, breaking down proteins during that fasted state. You've seen this slide before. There are 3 ways we regulate these pathways. One of it is by making the new enzyme from scratch. This is actually called de novo synthesis. And you'll see this term more. But de novo synthesis is synthesis from scratch. So that's where you actually go, the body goes into the cell and it turns off the synthesis of the enzyme. And we already know allosteric versus covalent regulation. Allosteric is where a molecule binds to the enzyme, alters its conformation, which is the shape of that enzyme, which is going to increase or decrease that enzyme's activity. So in other words, if you have an enzyme here, this is the enzyme, it's going to have an active site where it binds to its substrate, like right here, it's an active site. But it's also going to have another site where it's an allosteric site that can sit on the enzyme and turn it on or turn it off. So you either have an active site or an allosteric site, or you have both of them. The active site is where the substrate sits, whereas the allosteric site here, this would be the allosteric site. That is where a molecule will sit on the enzyme and inhibit it or enhance it, depending on what it wants to do. So in other words, it will, the molecule binds to the enzyme, alters the shape, increasing or decreasing the enzyme activity. That's allosteric regulation. Covalent regulation, that's what happens with insulin and glucagon. They modify the enzyme by its phosphorylation. So, you guys tell me, glucagon turns on a kinase or a phosphatase? A kinase. And so this kinase, what does it do? Does it phosphorylate enzymes or dephosphorylate enzymes? It phosphorylates enzymes. And that is covalent regulation versus insulin, then is the other one, and it's going to activate a phosphatase, and that phosphatase will take off the phosphate group. That's what a phosphatase does. It takes off the phosphate group, and that will turn on or off the enzyme. That's covalent regulation. So they modify enzymes in metabolic pathways with phosphorylation reactions, which turn the enzyme on or off depending on the enzyme. Are you guys thinking, why did I come back from spring break?
Yeah.
You're doing allosteric and covalent regulation still. All right. So we're going to get to the pathways, but that was just a little bit of review. So fats is energy. Keep in mind, fat is in a more reduced state than carbs. So I already told you guys what that means. Fatty acids are in more of a reduced state. What does that mean they have? More electrons. Fatty acids have more electrons because, again, fatty acids are like a 16 carbon fatty acid versus carbs just are 6 carbons. Glucose is just a 6 carbon. And so you have a lot more electrons that you can pull from fatty acids because they have a lot more carbons, which means they have a lot more electrons that you can take away. So greater oxidation, so they're going to be in a more reduced state, so there's going to be greater oxidation of fatty acids on their way to making carbon dioxide and water. So remember what happens to all those carbons? Same thing, they leave as carbon dioxide. So all those carbons will leave as carbon dioxide, and all those electrons will be moved to NADH and FADH2. So what does it mean to be in a more reduced state? Again, to be in more reduced state means there's more electrons in that molecule, more electrons. Here's the lipid pathways. These are the ones we're going to talk about in this class or in this lecture, except for cholesterol synthesis. We do talk about cholesterol synthesis, but that's going to be in the next lecture. But these are the ones in this lecture. We have lipogenesis, where we make lipids. Lipolysis, where we break down lipids. Beta oxidation is where we break down fatty acids to make ATP. And then the last part of this lecture is going to be ketogenesis, and that's where we make ketones. So similar to carbs, fed versus fasted states determine which pathways are active or inhibited. So the fed state of the adipocyte, blood glucose increases, insulin is released in the fed state. So in the fed state, you're going to build stuff. You have extra energy, and so that's what's going to happen. It's going to favor triglyceride synthesis. If you don't know what TG means, TG is always my abbreviation for triglycerides. So triglycerides are going to be synthesized in the adipocyte. That's going to be called lipogenesis. So genesis is the creation of lipids. Insulin is going to be behind this. Insulin dominates in the fed state. And it's going to, one, we already know this, it's the one that's going to help glucose enter the cell. But it also increases the availability and uptake of fatty acids by adipose tissue. So it turns on this enzyme. So, okay, let's do slightly better review here. Remember chylomicrons. Chylomicrons are made in what cells of the body? Made in enterocytes. Enterocytes are intestinal cells, enterocytes. And remember, where do they go? Do they go, chylomicrons, do they go to the lymph or the circulatory system? They go into the lymph from the enterocyte. They eventually make it into the blood. And from the blood, they're going to deliver triglycerides from the diet two tissues. So how do cells get the fat from chylomicrons? They do it via this enzyme, lipoprotein lipase.
Bless.
Bless you. They do it using lipoprotein lipase. Sometimes they just call it LPO. But lipoprotein lipase, remember what a chylomicron is. It's one of your lipoproteins. We're going to talk about your others later, like LDL and HDL. But it's a lipoprotein. Chylomicrons are lipoproteins. And so they need a lipoprotein lipase. Lipoprotein lipase is going to pull the fatty acids off of that triglyceride in order to get them into the cell. So without lipoprotein lipase, you can't actually pull the triglycerides from the chylomicrons. So again, these triglycerides, there's going to be this lipoprotein lipase that's going to pull the fatty acids. Oh, that's right, fatty acids from the chylomicron. Oh, I don't know how I did that, but I kind of like it. So that's what lipoprotein lipase does. And what activates lipoprotein lipase? Insulin. Insulin activates it. So without lipoprotein lipase, you can't really get the triglyceride, the fatty acids from the chylomicron. So you need lipoprotein lipase, and insulin is what's going to activate that lipoprotein lipase. And then you can store it as triglycerides in your fat tissue. Here's another lipase we're going to talk about in a little bit. It's called hormone-sensitive lipase. I'm just introducing it here. It's going to be dominant during the fasted state. So insulin activates lipoprotein lipase. In the fasted state, what do you think is going to activate hormone-sensitive lipase? Glucagon. So insulin will activate lipoprotein lipase. Hormone-sensitive lipase will actually be the one that's going to activate, glucagon is going to activate hormone-sensitive lipase, but that's going to be during fasted, which we'll talk about in a little bit later. We're going to focus now on the fed state. So in the fed state, the liver, by the way, the liver is a really important organ. It's got a lot of functions. One of them is nutrient metabolism. It makes sure that the nutrients go where they need to go in the body. So it's kind of this site that regulates nutrients, toxins, helps make things to deliver them to the body. So it actually gets a lot of the nutrients that we absorb from our diet. A lot of that's going to end up in the liver, and the liver is going to figure out what to do with it. So one of those things it's going to do is take up chylomicron remnants. So when the remnant, the chylomicron, has delivered its great stuff, its nutrients, its triglycerides to all of those tissues, it's going to become a remnant. And it's going to be endocytosed into the liver. So that's what's going to happen to the rest of the chylomicron. It's delivered all its packages of triglycerides to all of the adipocytes. Muscle cells might want the triglycerides because muscles like to use it for energy. And once it's done, the rest of the chylomicron, which is the remnant, will end up in the liver. And that's where the liver does not store lipids. If the liver stores lipids, that's called hepatitis. That's when the liver is dysfunctional. You do not want the liver to store a lot of lipids. It's called fatty liver. Fatty liver is not healthy, not in humans anyway. And so the liver will, what it's going to do is take those lipids and it's going to repackage them into VLDLs and send it back out to the blood. And we'll talk about that later. I know I keep telling you, we're going to talk about that later. But we will when we get to lipoproteins, which is going to be next week. So we're going to make some LDLs, some HDLs, and other things to put in the blood, or to put in the blood for the tissues. The liver is also going to be really important for making cholesterol. So it's the primary site for cholesterol synthesis. Your other cells can do it, but really the liver is the only one that does. The others do it, but in a very small way. And in the liver, cholesterol is going to be important for making bile. Remember, that's what one of the functions of cholesterol is to make bile. Another function of cholesterol is to make steroid hormones, your sex hormones. Vitamin D. Cholesterol is also going to be really important in cell membranes, as well as lipoproteins. So we'll talk a lot more about cholesterol next week as well. Actually, might be this week. All right, fasted state. In the fasted state, insulin levels drop. Thus, it no longer inhibits what enzyme? What enzyme did I tell you glucagon is going to turn on? Hormone-sensitive lipase. So in the fasted state, hormone-sensitive lipase is going to become activated. And that's going to be because of glucagon. So fatty acids circulate in the blood, bound to albumin, and are taken up by the liver or the muscles for oxidation by way of acetyl-CoA production. Are you guys starting to understand some of these sentences? Like, this was a very scientific sentence.
No.
Somebody said no. Free fatty acids circulate in the blood, bound to albumin, and are taken up by the liver or muscle cells for oxidation by way of acetyl-CoA production. Like, this kind of sentence is something that I just want you guys to eventually just understand without thinking about it anymore. I don't know if that'll happen, but.
Do those free fatty acids have to be short and medium chain?
So the thing with albumin, it does carry long chain as well, but only when it's been broken down from the adipocytes. So that's actually a really good question, because I told you from the diet, medium and short chain are carried by albumin. But when it's not from the diet, when your tissues, your adipose tissues are being broken down and those fatty acids are being released, that's when albumin will also carry the long chain fatty acids. So, and we will talk about that too. So this is just a heads up of things we're about to really dive into in a much more detailed way. And we're also going to talk about what happens to the glycerol backbone. Because remember, when we break down fatty acids, we've got, or triglycerides, we've got these three fatty acids that we take off. But what happens to this guy? This is the glycerol. And I'm going to tell you what happens to it. It's not as simple as we just throw it away. But this guy can actually be sent back to the liver. It can become glyceraldehyde 3-phosphate. Do you guys remember what this is from? What pathway do you see glyceraldehyde 3-phosphate? Glycolysis. And so glycerol will actually become glyceraldehyde 3-phosphate. And so you can do glycolysis with it, or you can actually put this back into the blood, take it back to the adipocytes, and the adipocytes are going to use it to make more triglycerides. So there's a few things you can do with G3P, but you can use it just to put it straight into glycolysis. So you can do glycolysis, or you can make triglycerides with G3P. So, all right. So we've talked about energy metabolism. We talked about fed versus fasted states, and I gave you a general idea of what's happening during them. Now we're going to focus on fatty acid synthesis. So what hormone is dominating when fatty acids will be synthesized? Glucagon or insulin? Insulin. So insulin is going to be dominating, and we're going to focus on this, and we'll talk about these guys next. So fatty acid synthesis is going to increase with a high-calorie, high-carb diet. It's going to decrease with a low-calorie or high-fat diet. Why is it going to decrease with a high-fat diet? Because you ate the fat. You don't need to make more if you've already eaten a lot of fat. So it just depends on the body's needs, ultimately. If the body needs it, it's going to make it. And it primarily occurs here, the cytosol, not the mitochondria. We'll be talking a lot about the mitochondria, but fatty acid synthesis takes place in the cytosol, primarily in the organ that is the liver. That is really where most fatty acid synthesis occurs, in the cytosol of the liver cells. And here's that NADPH. Where did we make, this is going to require, fatty acid synthesis, and we did talk about this very briefly last term. Fatty acid synthesis does require NADPH. Where did we produce it? We talked about this last term. Pentose phosphate shunt, or you can say PPP for the pentose phosphate pathway. But that's where we make, the primary way we make NADPH. And we need this. It is basically the reducing power. NADPH is the reducing power we need to make fatty acids. Because remember, to break down fatty acids, we oxidize them. We get rid of the electrons. We take their electrons. But if we're making fatty acids, you got to add electrons. Where do we get the electrons? We get them from NADPH. So it is going to reduce. It's going to give the electrons back to the fatty acid. And that way, you have the fatty acid that has, well, you wouldn't be able to make it without NADPH because they need electrons. All right, here you go. These are the steps you need to memorize. And I'll say it's better than, where is it? This. I wanted to show you guys my iPad, but you see it. This is fatty acid synthesis, or at least 3/4 of it. Do not memorize this. I have a lot of images in my slides that I tell you guys, I want you guys to see it, but don't memorize it. What do I want you guys to know? I'm going to explain it, but ultimately, this is the pathway that I want you guys to know. I've taken out a lot. But this is the gist of it, and that's what I really want you guys to get, not the details of where exactly things are moved around, but the gist of it. So fatty acid synthesis, the first step. I'm going to tell you all the stuff that was missed last term. Okay, the first step is going to produce a malonyl-CoA. Do remember malonyl-CoA. It is. There's some M words going to be later on when we talk about cholesterol synthesis and some other stuff. Malonyl-CoA is specific for fatty acid synthesis. So we're going to produce a malonyl-CoA, and this is the first step here. And it is going to be catalyzed by an enzyme you have to know, and this enzyme is acetyl-CoA carboxylase. It's telling you what it's going to do. Carboxylase, you're going to be adding a carbon here. So acetyl-CoA carboxylase, we are going to start with acetyl-CoA. Acetyl-CoA. How many carbons does acetyl-CoA have? 2 carbons. So acetyl-CoA is a 2-carbon molecule. We need to make a 3-carbon molecule that's malonyl-CoA. So malonyl-CoA is a 3-carbon. So we went from a 2-carbon to now a 3-carbon group, which is malonyl-CoA. And we do need ATP. We're building something. That's why you only want to do it when you have extra energy lying around, when you're in the fed state. So we're in the fed state right now. And this enzyme is the major enzyme that you need to remember. And we'll talk about how it's regulated in a second here. But that's the first step. You're going to make malonyl-CoA simply by putting a carbon onto acetyl-CoA. Just a heads up, guys. When we make cholesterol, we start with acetyl-CoA. When we make ketones, we start with acetyl-CoA. When we make fatty acids, we start with acetyl-CoA. So acetyl-CoA is that starting material there for all those. Just giving you a heads up because it'll help you kind of remember some of these. But we start with acetyl-CoA, we need to add a carbon, and now we have malonyl-CoA, which is a 3-carbon molecule. So here's the first step, acetyl-CoA, it's right here. We use this enzyme, acetyl-CoA carboxylase. It's going to add a carbon. So this is bicarbonate. It could have used carbon dioxide. We just need a carbon. And so it's going to add a carbon here, and now we've got a 3-carbon malonyl-CoA. Very first step. Step 2. This is step 2. This picture right here. But I'm going to tell you exactly the takeaway from that. Step 2 of fatty acid synthesis, it's a multi-enzyme complex, which is why there's this really long diagram showing that how it works. But all that's going to happen is this malonyl-CoA is going to condense. That means it's going to covalently bond to, it's going to add itself to, and with an acetyl group to form a 4-carbon butyryl, which I don't care if you remember that. But essentially, what I want you to understand is you have malonyl-CoA that's going to condense with another acetyl group. And you're essentially going to keep it, I should back up. You're also going to need NADPH in this reaction. You're going to need NADPH in this reaction, which again we get from the pentose phosphate shunt pathway. And this, I do want to show you this picture here. Sorry, I'm going to give you guys a break. So this malonyl-CoA, we use NADPH here, and we're going to use another enzyme called fatty acid synthase, and we're going to essentially take malonyl-CoA, sorry, it's blocked here, plus acetyl-CoA, and we're going to keep adding these two together over and over and over until we end up with a long fatty acid chain. So this one is palmitate. Palmitate is 16 carbons. It's the most common one. So we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 carbon fatty acid. Palmitate is a very common one. It's the primary product of fatty acid synthesis. So this is the enzyme right here, fatty acid synthase. And it is a multi-enzyme complex, and I'm going to show you that in a second. I do want you to at least visualize it, even though all I want you to remember is fatty acid synthase. That's the one that's going to take malonyl-CoA and condense it with another acetyl-CoA, and you're going to keep building, adding more and more acetyl-CoAs until you finally get the 16 carbon palmitate. So that's what step 3 is repeat. You're going to just keep repeating, condensing with another malonyl-CoA to form a 6 carbon. Keep going. You can form upwards of 22 carbon units. Actually, upwards of 24 carbon units, technically. And if your body needs an unsaturated, remember unsaturated, you got to take away those hydrogens, you actually have what's called desaturases. So these are desaturases are enzymes that are going to take away those hydrogens in order to put a double bond between the carbons. So it creates a kink in that chain. Remember, if you're making an unsaturated fatty acid, it means you have a kink in the chain. It's because you have a double bond between the carbons. And that's just going to be done using, and you just have to remember desaturases are what's going to make the double bond between the carbons and desaturate the, and make the saturated fatty acid an unsaturated fatty acid. So, I'm going to show you this fatty acid synthase really quickly. Oh, actually, I'm gonna stick with your slides. I'm gonna go back and just talk about this first one, acetyl-CoA carboxylase. So remember, this is the regulated enzyme. Regulated enzyme. So this is the regulation you need to know for this pathway. It's the only enzyme that's regulated in this pathway, or regulated significantly, I should say. And it's regulated by both covalent and allosteric regulation. So remember what it does. It actually is what's going to make malonyl-CoA. It's what's making malonyl-CoA the very first step. Covalent stimulation, remember when the fed state. So what enzyme is going to activate it, or what hormone is going to activate it, you should already know insulin is going to activate it. And glucagon will inhibit it. Because when the fed state, insulin, you should already know will activate it, and you should already know because glucagon's a fasted hormone, it's going to be the one to inhibit it. And that's what happens. In covalent regulation, it's stimulated by insulin. Remember, insulin stimulates a phosphatase, removes phosphate groups, and that makes acetyl-CoA carboxylase active. It is inhibited by the hormone glucagon, which again happens in the fasted state. Glucagon activates a kinase. Kinase phosphorylates enzymes, and when you phosphorylate acetyl-CoA carboxylates, you make it inactive. That's covalent regulation. So we're either phosphorylating or dephosphorylating it. In this case, dephosphorylating it makes it active, and that's what insulin does, or insulin activates that pathway. And then allosteric regulation, this is the one, again, if you understand fasted versus fed and the molecules involved in that, then you know that citrate indicates excess energy. Where do we see citrate? What is the pathway that we see citrate? Krebs cycle, TCA cycle, whatever you want to call it. That only is activated when you have lots of energy. So if citrate is around, that means you have excess energy. So citrate can go sit on acetyl-CoA carboxylase. So if we have acetyl-CoA carboxylase, citrate will actually go sit on it and turn it on. It'll actually activate that enzyme just by sitting on it. And of course, just like we have an activation, which is citrate, we also have an inhibition. And that inhibition is really easy to remember. It is palmitate itself. Palmitoyl-CoA is just another way of saying palmitate. Palmitate. So if you have already a lot of palmitate, it can go back, sit on the enzyme, and turn it off. And that's allosteric regulation. And this is the picture from your textbook. It's showing the same thing. Insulin activates a phosphatase. The phosphatase will remove the phosphate group, making acetyl-CoA active. Versus if you're in the fasted state, glucagon is going to be dominant. And in this case, it's going to activate a kinase, which is going to put the phosphate group back on, and that makes it inactive. That's just the pictures from your textbook showing the same thing, but this is covalent regulation. Covalent regulation. I also want to mention, just really quickly here, you'll see this again, because a lot of students were confused by this last term as well. CoA, whenever you use a fatty acid, you actually have to add a coenzyme A to it. So don't be, it's just the way that in order for the cell to move fatty acids around the cell, you actually have to add a coenzyme A, a CoA. So palmitate and palmitoyl-CoA, like I said, I use them interchangeably. It's basically palmitoyl-CoA is just a palmitate with a coenzyme A added to it. And coenzyme A is just something you have to add to these guys in order to get them, to move them around the cell.
Excuse me.
Yep.
So a question as far as the synthesis of steps that we just went over?
Yeah.
Yeah, right there. So you get to step 3. You made the 4 carbon from malonyl CoA condensing with acetyl-CoA. Add the carbon, now that's 4, right?
Yeah, technically, so acetyl-CoA is a 2-carbon, malonyl-CoA is a 5-carbon, so technically you end up with a 5-carbon, but you lose a carbon in that, so you end up with a 4-carbon.
And then it just adds that again with another malonyl-CoA.
You get another malonyl-CoA and condense that as well, and you keep doing that. So yeah, you repeat condensing with another malonyl-CoA, and I'm actually going to show the picture and kind of try to walk you through that diagram that's from the textbook. I'm not a huge fan of that big diagram.
So when it says 6 carbon, where do we lose it?
Every time you add a malonyl-CoA, which is a 3-carbon, you lose a carbon in that addition of that malonyl-CoA. Yeah. So you can see here, you actually lose these carbons as carbon dioxide here. And so every time you go through a round of adding another malonyl-CoA, you lose another carbon. And so, yeah. But
the whole purpose of the synthesis is to take a fatty acid that is saturated and desaturate it.
nope, you're just making palmitate. So you're starting with, really the starting material is acetyl-CoA. It's always acetyl-CoA. And even to make malonyl-CoA, what did we use? We used acetyl-CoA. And so essentially, you're just using acetyl-CoA to keep adding 2 carbon units until you eventually make a fatty acid, a 16 carbon fatty acid, or whatever size you want. So really, every round of this is adding 2 more carbons to the chain until you eventually get, for example, a 16 carbon palmitate, or you can make a 20 carbon, a 22 carbon. Those desaturases down here, that's only when so desaturases, these guys are only if you want to make an unsaturated fatty acid. You have to throw in a desaturation enzyme that's going to desaturate that fatty acid. So this palmitate is saturated actually. It's a saturated fatty acid, but if you wanted to make it unsaturated, you would just add in here a desaturase enzyme, and your cells can actually make a double bond here and actually get rid of these hydrogens, so you end up with an unsaturated fatty acid. So your body will do that too. It just depends on whether your body needs a saturated fatty acid or an unsaturated one with what it's making. And sometimes, remember, these fatty acids could be used to make cell membranes, not just for triglyceride synthesis. So sometimes it needs to make the fatty acid unsaturated. Oh, I don't think I mentioned, cell membranes tend to be unsaturated fatty acids, whereas your triglycerides are oftentimes saturated. You don't have to remember that kind of detail, but it just depends on what the cell needs as to whether it decides to make an unsaturated or a saturated fatty acid. And here's the allosteric regulation that I just talked about. Remember, citrate is going to be what turns on acetyl-CoA carboxylase, whereas palmitoyl-CoA, again, palmitate, is going to sit on the enzyme and turn off acetyl-CoA carboxylase. But do remember the enzyme that's regulated. That's the most important one. The other enzyme is also super important. But there's only one other enzyme you guys have to remember. Fatty acid synthesis only has 2 enzymes you need to remember. I guess 3 if you count the desaturase. The first enzyme is acetyl-CoA carboxylase, and the second enzyme is fatty acid synthase. So it should be easy to remember. And here's another little detail. I'll be honest with you, I don't think I care very much about this detail. So I probably won't put it on the exam, but I'm going to tell you anyway. Acetyl-CoA, most of it's in the mitochondria, guys. And so we got to get it out of the mitochondria. We can't just move acetyl-CoA out. We actually have to turn it into something. We have to turn it into citrate. Citrate can leave the mitochondria, and in the cytosol, it can be turned back into acetyl-CoA. It might be important. You might see it on the board. I don't know. But I threw it out there. But it's not something you'll probably see on the exam. I'll say that too. But I have told you about it, so I've done the job. All right, so we did talk a lot about this first step. I just want to go over a little bit more about the fatty acid synthase, because it is a little confusing. This malonyl-CoA has to condense with the 2-carbon acetyl-CoA to form the 4-carbon molecule, and then we need to repeat. So this is the same slide. I just now want to talk about this. And I really stressed my students out last term because I didn't really make it clear that you don't really need to know this. But here, this green guy, I do want to at least tell you what these are doing. This green guy is fatty acid synthase. And I made the unfortunate thing of where just I drew it out too. This is a really interesting enzyme. And if you are biochemistry students, I would want to spend time on this, because it does a lot of complex, interesting things. But I'll tell you what you really need to know after this question. Yeah. Yes. No. I'm having so much fun. But yeah, let's take a break. Please, let's take a break. Okay. I'm gonna put up quickly. Sorry, guys.
Oh, let's go.
You guys inspire me, and I can just go on. Okay.
Are you acting?
I'll stick it out then.
No.
I thought that you had a plan.
I don't have a plan.
It looked like you were confidently just going through, and I was like, all right.
I don't know about confident, but I was going through it.
You were going. I was like, all right, maybe she's gonna. Maybe there's
It's gonna
some.
be really exciting.
There's something at the end of the road.
No, it wasn't worth it.
It's okay. Make sure you take it.
There's another one. So I definitely noticed the regulations. So this regulation is really short. Their regulations for fatty acid synthesis. So citrate, which is like on, this is going on, sitting on it, they can sit on it. That's really.
I'm trying to make sense.
No. Well, insulin and glucagon down here. So insulin, this is covalent regulation. So the difference between these two.
Are you flirting as well?
No.
Are you sure about that?
Hello. I'm not flirting. I have this is important things to do.
Advancing. Oh, really? You at least tell me which protein or which enzyme breaks down chiromycins.
Because you remember the end product of this.
You do
Is to make it blood. Since you
I was
already have
Which protein or which enzyme breaks down chiromycins?
yeah, it's the primary. And so when it's already accessible, then it'll just go back. It's like a negative feedback. Go back and sit on the enzyme, trying to make it turn. This is the. This is what this enzyme does. This is the
I actually don't know if I can.
catalytic reaction. So it's
Is everyone just exhausted? What's going on? Why is everyone fleeing?
because the process is two carbon coax adding a carbon.
Oh, yeah.
With the carbons are one.
I'm like that. If I was like 10% more awake, I could probably understand what's going on.
No, that's I usually just talk about the tree by add the carbon. No, this carbon stay. These two carbons stay. Add another carbon. That's there's three hydrogens. Yeah. Well, this is the one where yeah, so no, it is not. It's
I went over made an observation in my exercise physiology class last semester of undergrad, so that is somewhere still in my brain.
okay.
He's gonna go over the fibers again in the
Yeah,
next
this
one.
one is just telling you how so
Maybe I don't understand Bermuda's PowerPoint. That's where I'm concerned.
yeah, the regular.
Next question.
Oh,
I made them.
Oh, no, yeah, for sure.
I did them. I already started back to the first two. Like if you go back to lecture 1, it already has it. Because another student made. It's this one, yep. And I will put it on the other ones. That's a minute. Just order other students last one again, so I don't have time to wait for them. But yeah, it's gonna be between them. So if you go back to the, I don't know when you download this lecture, but if you download in the first lecture. Yeah. Well, it is, but it does. I don't have
It's nice that I can do that. It's like
Yeah.
So there's like 1 or 2 minutes of
How are you?
You're just sitting in this sad, like, I can't believe this is my life, now it doesn't even come for you.
you guys stick it out, though.
I just,
Stick it out. Still like it's still open for 3 more hours.
we'll have to stay open. I can stay. Yeah, well, Saturday after grace started, please. I can try. Like, this is I feel like
Yeah, I think it's for
So really, this is just trying to make sure you're using the difference between anaerobic fat oxygen and anaerobic.
at that point, don't come back to it. One to 4:30 is crazy.
And then we have lypolysis is anaerobic. And it's tracking when we do that, there's no oxygen present. We just take
That's crazy. One to 4:30 is such a crazy lunch. That's a crazy lunch. Schedule me in the morning. See, and those people, I can make a special exception because everyone take a lunch, right? Come here for lunch break. If those people that can come at 8, they can come during the lunch break. And your little 3-hour, 4-hour little break that you got, you can spend at least 30 minutes. I'm sending blessings your way, man. I hope you get the right rest and you got the ability to do that. 4 hours? Bro, 4 hours for a break. It's basically 4 at that point. Because by the time you start running up, it's going 4 hours. Sure. Okay, if you don't play much of sleep, how come you didn't get a full 8 hours rest?
So when
About ladies about 8:30. I would be in the bed by what, 10:30?
And then you what? You open your practice at what, 8 o'clock?
that's where.
What time? Damn, 10, 11, 12, 1. Damn, you're not even open. Who are you hitting? What's your target audience? God, you were open for 2 hours and then it don't even make sense. 6 people waiting for you. That's crazy. At that point, you'd be just open up like 4 to 8. That's so pointless. That's one of the things that most he's gonna be chilling in the office.
Have you heard this TikTok sound?
This is like
It's so fucking funny.
My favorite right now
Guys, just like, not again.
It's okay, it's okay. So basically we're going over fatty acids.
Okay, I got that.
So where did it start with? Which makes Shout out. No.
No, if you did, I would not be here.
Why? What do you have after?
I was just wondering because this is like,
Because she thinks this is horrible and we have to say,
like for me too.
Yeah.
So bad.
I don't know why they didn't like our
Then we have a 4-hour break in between our 7 a.m. We have 7 a.m. to 9.
You miss 70. I mean, I do too, but I don't.
But it goes after our 7 a.m. So from 9 to 1 we have a break. Instead of just bringing all these classes up.
You know what? I have a 4-hour break too. Really? I have a 7 a.m. and 9 a.m. but I'm 11 to 3. Like, well, I'm doing other stuff. But yeah, part of me is just like, let's get this done early.
I'm always like, I really get it done.
And you guys don't have anything either.
We always have
I know. I always have something to do. Yeah.
Clubs, we got clubs.
Okay, good.
Can you come to one of our adjusting clubs?
Oh yeah,
Do I get adjusted?
yes, of course. Yeah. But don't let God get you. I got you.
Wait. Do you guys actually do the adjusting?
No, not really yet. No, that's good.
we're like,
Not that it's concerned.
she's like, this never mind.
Maybe you're wrong.
Yeah,
Maybe a little.
I will not get you.
Not sure I want to be a guinea pig. And like,
Who cares?
No, I'm actually a good adjuster. I'll get you right.
You're bad. You're bad.
no, is there like other students, like older students?
Yeah.
No, and also there's like people that are not students, right?
Is there chiropractic?
Yeah, there's chiropractors. There's people. It depends, and they can come and teach you, and then they go around. Yeah.
Great.
Like Dr. Thornhill does it for Thompson.
Yeah, for Thompson Club, Dr. Thornhill's there.
Really?
Yeah.
The advisors have to be there for like.
Those are for the on-campus ones. There's off-campus ones where the students are allowed to, or the students adjust. No one doing it.
Yeah, you're about to say aloud.
You can't really tell me.
Scratch that.
Yeah. Because we can't do it here till we take full spine, right?
Yeah.
I think.
Yeah, even then, we would have to be watched over by adults.
But yeah.
Yeah. And that makes sense.
Do you have
Yeah,
an intern?
I don't think we get student interns, but I
For
get a
sure.
student.
It's definitely up to
Hell
you
yeah,
if you get a student
free
intern.
chiropractic care here.
Yeah. You get free, yeah, exactly.
Oh, that's
dumb
what
'cause she doesn't want you to
someone
adjust.
who literally just
Adjust you. Yeah, I mean, it's a student, but it's an upper quarter student that should know. Understandable.
Okay, I'm going to tell you what you need to know about this fatty acid synthase, which again, like I said, it's a multi-enzyme complex. It's got a lot of capabilities. But what's really happening essentially is we're taking a malonyl-CoA and we're condensing it with another 2-carbon acetyl-CoA group, and we're going to then repeat that over and over with malonyl-CoA. And I'm going to show you that in this. I'm going to try to show you that here. So, kind of keep it simple for you guys. So I don't. Here's the fatty acid synthase. So that's the name of the enzyme, and it does a lot. As you can see here, it does these all the way through step 7 here. It does all of these chemical reactions. So in this case, it's taking. So we already made step 1. We already made malonyl-CoA. That was with acetyl-CoA carboxylase. So here's that malonyl-CoA that's coming in, and it's going to condense with the acetyl-CoA that's already on this fatty acid synthase. And so that's what happens. We lose a carbon dioxide and we end up with this 2 carbon attached to that original acetyl-CoA that's there. Like all this detail that I went over with my students last term, and I'm trying to think like, okay. Oh, I do want to point this out. Here is where NADPH is. So remember, we do have to reduce the fatty acid that we're making. By reducing it, we're putting the electrons, and the electrons are in the form of those hydrogens there. So here's a hydrogen. These hydrogens are our way of, or your body's way of reducing that fatty acids, because those hydrogens contain the electrons. And so they take the hydrogens, those electrons and those hydrogens there are going to be added to the growing fatty acid chain. And so what we end up with, NADPH becomes NADP+, when it loses its electrons. So NADPH is oxidized to become NADP+. It loses its electrons, and the electrons were put on the growing fatty acid chain. So the fatty acid chain is being reduced, and NADH is being oxidized to NADP+. So again, oxidized is when you lose electrons, reduced is when you gain electrons. So NADPH is the reducing power for doing fatty acid synthesis. I think that is one of your quiz questions, and even probably an exam question, somewhat like that. Because NADPH is a really important molecule for making these fatty acids. We need it in order to put the hydrogens, those electrons, onto the fatty acid. So the fatty acid synthase itself, this really cool enzyme, does a lot of things. It takes this malonyl-CoA and it condenses it with another acetyl-CoA, and here we have repeat. So in this 2, 3, 4, 5, 6, 7, we just keep repeating these first 7 steps over and over and over. And you don't have to memorize these 7 steps. This is what I want you to know. What fatty acid synthase does is it's going to take, so here you have acetyl-CoA, which is a 2-carbon molecule, and it's going to condense with a 3-carbon molecule, which is, so this is acetyl-CoA. Acetyl-CoA here, and this one's malonyl-CoA. Malonyl-CoA. And fatty acid synthase is going to take these two, condense them, and it's going to make a 4-carbon. 4-carbon. And so we're actually going to lose 1 carbon here. But it's going to do this over and over and over. So it's going to, so this is the fatty acid synthase that's doing this. And then it's going to do it again. It's going to add another malonyl-CoA, which is a 3-carbon, and it's going to take that. So again, fatty acid synthesis is doing this, and it's going to make, so we had originally a 4-carbon, 2, 3, 4, and now we're going to add 2 more carbons to this, and so we now have got a 6-carbon, and we leave just another carbon dioxide. And then we're going to do this again. We're going to take another, we're going to add another malonyl-CoA, which is a 3-carbon. So we just keep adding more and more malonyl-CoAs over and over, and this happens though in all of these 7 steps. So, but essentially all it is just us taking the original acetyl-CoA that's attached to the fatty acid synthase here. So there is an acetyl-CoA attached to the fatty acid synthase. You can see the acetyl-CoA here. It's attached to the fatty acid synthase, which is the green thing here. The green enzyme is fatty acid synthase. And then we just keep doing this over and over, adding another malonyl-CoA. So here's adding another malonyl-CoA. If we were to do this, they only show it twice. So they show all of these steps twice. So we need malonyl-CoA, but we also need to make sure to remember we need NADPH. Hopefully I have that written down here. Do I have NADPH? I do not, so let me write it in. this is going to need, okay, it's erasing, need NADPH. That's where the enzyme that needs NADPH is fatty acid synthase. And all it does is take malonyl-CoA and condense it with a 2-carbon acetyl group on the fatty acid synthase to form a 4-carbon molecule. And then it does it again, it repeats. It takes the 4-carbon molecule that we just made, condenses it with another malonyl-CoA to form a 6-carbon. And then it's going to repeat. It's going to take the 6-carbon, add another malonyl-CoA, and we're going to end up with an 8-carbon. And then it's going to repeat. It's going to take the 8-carbon, and it's going to add another malonyl-CoA, and it's going to then make a 10-carbon, and it's going to repeat. It's going to take another malonyl-CoA, add that to the 10-carbon we just made, and we're going to end up with a 12-carbon. That's what fatty acid synthase does. It just keeps taking these malonyl-CoAs and adds them to the growing fatty acid chain. So we add to the growing fatty acid chain 2 carbons at a time. Because remember, every time we lose a carbon dioxide, malonyl-CoA is a 3-carbon molecule. So every time we add another malonyl-CoA to the growing fatty acid chain, one of those carbons will be lost as carbon dioxide. But we keep doing this to produce upwards of 22 carbon chain fatty acids. Yes?
You said NADPH is the reducing power. What is NADP plus?
It is what happens when NADPH does its reduction activity. When it gives the electrons away, it becomes NADP+. So NADPH is the reduced form. When it gives up its electrons to the growing fatty acid chain, it becomes oxidized. And the oxidized form of NADPH is NADP+. So NADP+ is what it becomes when it loses its electrons. But it's just adding malonyl-CoAs over and over again until we end up making the size of a fatty acid that the body is trying to make. And it really depends on the cell, whether it's making a 16 carbon, 22 carbon, it just depends on the needs of the cell. So, but that's what it's doing this whole time. It's just taking malonyl-CoA, adding it over and over to produce. And the end result, like I said, it can be a longer chain, but usually it's palmitate. Palmitate is the most common product of fatty acid synthesis. Yeah. Nope. It's so, well, I would say it's very different because that uses like glycogen and we don't want to get into that. It is. You can compare it to it in that it's growing something. And so to grow, it has to keep adding more and more units. In this case, the units that keep being added is malonyl-CoA. But remember, how did we make malonyl-CoA? What was the actual starting material in order to make malonyl-CoA? Acetyl-CoA. So acetyl-CoA, all we have to do is add a carbon to it, and that's what acetyl-CoA carboxylase does. The enzyme even tells you what it's doing. It's carboxylating acetyl-CoA. It's adding a carbon, and that's the product is malonyl-CoA, which is a 3-carbon. And then fatty acid synthase takes that malonyl-CoA that we made in the first step and keeps adding it over and over to the fatty acid chain. And so every time we add 2 carbons to the growing fatty acid chain, and we lose 1 carbon dioxide every time we do that. And so technically, even though malonyl-CoA is a 3-carbon molecule, we really remove one of the carbons and we just add 2 of those. So we add the acetyl-CoA. We're actually growing the fatty, I don't want to confuse you, so I won't say that. I'm walking that one back, just ignore that last sentence. But that, in a nutshell, is fatty acid synthesis. So you really have 2 enzymes to learn, and the fact that fatty acid synthase requires NADPH. Yeah. Oh yeah, so this image, again, what fatty acid synthase is doing, so we start with this first. Fatty acid synthase actually has 1 acetyl-CoA already attached to it. And it just adds another malonyl-CoA, we lose a carbon dioxide, and we end up with a 4 carbon. Then we repeat. We add another malonyl-CoA, or fatty acid synthase does, we lose a carbon dioxide, and then we end up, not with a 4 carbon now we have a 6 carbon. And then we repeat. We add another malonyl-CoA, and fatty acid synthase will just add 2 carbons to that, so we go from an 8 carbon to now we're gonna end up with a 10 carbon. So it just keeps doing that over and over. That's what it was, it's just repetition. So fatty acid synthase, this picture here, notice that fatty acid synthase already has, this is an acetyl-CoA right here. This is an acetyl-CoA. So an acetyl-CoA is already attached, so now we just have to add a malonyl-CoA to it.
I have a question.
Yes.
It says fatty acid synthesis, is that the same thing as palmitate? Is that the same thing?
Fatty acid synthesis is synthesizing fatty acids. Palmitate is one type of fatty acid. So palmitate is the most common fatty acid that we're trying to make. Your body will make all sorts of fatty acids, but the most common one is palmitate. So just get used to knowing palmitate is a 16 carbon fatty acid. It's probably good to know that one because it's just the most common one. So there's some fatty acids that will have you remember, like palmitate. 16 carbon fatty acid, it's saturated. 16 carbon saturated fatty acid is called palmitate. And if we want to desaturate palmitate and make a double bond between those carbons, what enzyme are we going to use? A desaturase. So again, and that just depends on what the body needs. If it needs a desaturase, it will use that to make it unsaturated. And here's the NADPH. We actually have 2 ways of making NADPH. Yes.
Palmitate is a 16 carbon unsaturated fat?
Saturated, not unsaturated. Yeah. And there are 2 ways that we make NADPH. I think it's important, perhaps, that you know both. But one of these you already know. The pentose phosphate pathway from last term, that's how we make most of your cells make NADPH that way. But there is another way we can actually make it when we convert pyruvate to oxaloacetate. So there is a second way, and that's showing you here. Your body can actually take oxaloacetate. Oxaloacetate can't leave the mitochondria, so first has to be converted to malate, and then it's going to be converted to pyruvate. And here's where we make NADPH. Okay, it's in my slides. It doesn't hurt to know it, but really the primary way and the way that this is the way that you really want to remember. Where do we get NADPH? We make it from the pentose phosphate pathway. So the other one, you probably won't see on my exam. It doesn't hurt to know it because you might see it elsewhere, but this is the way that I really want you guys to remember. This is where we get NADPH primarily. I have no idea what this slide is. Oh, great. This is actually showing you how we get, this is showing citrate and how citrate can go out and make acetyl-CoA. Remember that I told you didn't have to know that. And it's also showing you how we can actually take pyruvate, or we can take malate from pyruvate, and I make NADPH. It's not I don't find this slide very interesting. It does show you how everything's kind of hooked together. So NADPH is actually, these pathways flow together. So I talk about all these pathways as if they're separate. I guess I do like this picture in that it's showing you everything is actually just this cooperative dance going on in the cell of sharing intermediates in order to help make various things. It's a beautiful thing what the cell does. But I think this picture might be a little, feel free to look at it. It does go into a little bit more detail, but I'm gonna just skip it. So you can ignore the information. Everything, the information you need is in my other slides. That's a good one if you are looking for more detail. All right, so now we've made fatty acids. What are we gonna do with them? One thing we can do are to make membranes, so we can make phospholipids, but the main thing your body's gonna do with those fatty acids is gonna build triglycerides. That's gonna be the main thing it's gonna do. And so that's what we focus on, building of triglycerides. So synthesized fatty acids can be oxidized for energy, or they can be used to synthesize various lipids like your triglycerides, which are just gonna be stored in your adipocytes, or used for beta oxidation, oxidized for energy. It is time to go. Let's get out of here.